In this video, I will describe some of the hormones involved in the digestive system. We'll start our story up here, the stomach. Even before you consume any food, the vagus nerve is going to trigger gastric juices to be secreted from the gastric glands and motility of the muscularis externa, the 3 layers of muscle within the stomach wall. And essentially, this is going to prepare the stomach for ingestion of food. Generally, this first stimulus is going to occur when you think about food, when you smell food, even when you taste food. Anything before food, the bolus here, enters into the stomach lumen. As soon as the bolus is swallowed and enters the lumen of the stomach, it is going to change the environment of the stomach, which was before only made up of these gastric juices. The food is going to increase the pH of the gastric juices. Generally, the food is a much higher pH than the very acidic stomach acid. Also, before the stomach was empty, and now it is more full, so there's going to be stretch that's detected on the walls of the stomach. Okay. This is going to stimulate a couple things. First of all, if you eat a big enough meal and you fill this entire stomach, there's going to be more stretch on the fundus. That is going to inhibit the release of ghrelin into the blood and therefore inhibit your appetite. Of course, this takes a little bit of time, and you're not really going to feel full or not have an appetite for quite some time. Okay. The physical stimuli, food, increase in pH, and stretch, are going to be detected within the wall of the stomach, and ultimately this is going to trigger the release of a hormone called gastrin. This is the one hormone that is going to increase the activity of the stomach. Gastrin directly increases the motility of the stomach, therefore mixing the bolus together with the gastric juices and forming a fluid called chyme. Gastrin is also going to feed back and increase the secretion of substances from these gastric glands. This includes stomach acid, or hydrogen ions and chloride ions together, enzymes such as the inactive pepsinogen, and other substances like intrinsic factor. Gastrin is, of course, a hormone, and therefore it is released into the blood as opposed to being released into the lumen of the stomach here. Okay. Just for clarity, this blood vessel, going all around the digestive system, is not actually in this shape. It is merely drawn in this shape to show you that there are substances going into and out of the blood. Okay. Once the chyme has been digested enough in the stomach, small spurts of chyme are going to pass through the pyloric sphincter and into the duodenum, the first part of the small intestine. Now, once again, the chyme, filled with food as well as the gastric juices, is going to change the environment of the small intestine. The small intestine is not robust against a really low pH. And so, first and foremost, we really need to get that pH up so it doesn't damage the small intestine lining. Also, the chyme is filled with a lot of nutrients, and so we need to digest those nutrients and ultimately absorb them. And so, there are cells within the duodenum, so embedded within the lining, in fact, of the duodenum, that detect different substances within the chyme. Okay. First, we have lipids. Lipids are detected by a particular type of cell, and in response, they are going to secrete CCK, or cholecystokinin, into the blood. CCK has several functions. One is that CCK triggers the acinar cells, the glandular cells, exocrine glandular cells within the pancreas, to secrete enzyme-rich pancreatic fluid into the main pancreatic duct and ultimately into the lumen of the duodenum. So essentially here, the problem is too many of this type of nutrient. The solution is to break down the lipids with enzymes that only come from the pancreas. In fact, the pancreas is the one organ that actually synthesizes enzymes for all 4 biomolecule classes, as opposed to everything else. everything else in the GI tract, in fact, that does not, right? It's always missing something. Okay, cholecystokinin is also going to be important to the secretion of bile. Again, CCK was released in response to lipids, and so CCK is going to fix the lipid problem in particular by triggering the contraction of the gallbladder and therefore the release of stored bile. CCK triggers the liver to also release some of its bile, which is synthesized in the liver, down this common bile duct, through the hepatopancreatic sphincter, and into the duodenum. So now we can fix this lipid problem with not only bile, which is going to emulsify the lipids and make them, a little bit smaller and easy to digest, and the enzymes to actually break those bonds. And again, that is through triggering the gallbladder and the liver to release bile into the small intestine. CCK also acts within the hypothalamus. In fact, it is an anorexic signal. That is, it is going to reduce the appetite because, of course, we already have fatty food in our GI tract. We have enough energy to be sustained. The next stimulus is acid. Again, chyme is super acidic. Most of the enzymes from the small intestine as well as the pancreas function best at pH of about 8, not 1, 2, or 3 like in the stomach. And so acid is going to be detected by a different type of cell within the duodenal membrane. In response, these cells release secretin, and secretin triggers the pancreas to secrete buffers, right? Specifically bicarbonate-rich pancreatic juice. Okay, so these buffers, again, are secreted through the hepatopancreatic sphincter into the duodenum and ultimately can increase this pH. So secretin has fixed the acid problem with buffers to increase the pH. Okay. Glucose is detected by quite a few different cells. Glucose triggers the release of glucagon-like peptide 1, or GLP-1, which triggers the endocrine cells within the pancreas to release insulin. The idea here is that if glucose is in the lumen, it is soon going to be absorbed into the blood. When that's the case, there's going to be a blood sugar spike, and the only way to fix that is with insulin. The fastest way to fix that is with insulin. And so, as soon as glucose is detected, before even it gets down lower into the small intestine where it can be absorbed, it is going to trigger the release of insulin. Similarly, glucose is going to trigger the release of glucose-dependent insulinotropic peptide, GIP. Used to be called gastric inhibitory peptide, but it turns out that CCK, secretin, GLP-1, GIP, and this next one we're going to talk about, VIP, all of them have the ability to inhibit the stomach. So all of these intergastrones inhibit the release of gastrin and stomach activity in general. Okay, and essentially the idea here is that if there is food, if there's lipids and acid and glucose and all sorts of other things in the small intestine, we need to make sure that all of that stuff gets through before the stomach should start churning and squeeze past a little bit more chyme. Okay, so once there aren't more lipids and acid and glucose and proteins within the duodenum anymore, these hormones stop being secreted. That reduces the negative feedback up here on the stomach, and therefore the stomach increases its activity again, squeezes a little more chyme past that again, adds lipids, acid, glucose, etc. And this whole cycle starts all over again. Okay. So back here, glucose triggers the release of two different hormones that signal to the B cells, or the beta cells of the islets of Langerhans, or pancreatic islets, to release insulin into the blood. This is going to be very important here soon. Okay. Why not secrete just one? Well, each one of these has other actions as well, and those actions are different. Okay. First of all, GIP is going to trigger an increase in bone deposition. Okay, so building the bones right as you are eating and after you're eating a meal. Therefore, immediately depositing all of those nice nutrients that you just digested and absorbed into a structure that can use it. GIP also increases the formation of adipose, so the storage of fat within your fat cells. GLP-1, glucagon-like peptide 1, this is also going to be an anorexic molecule, so it's going to influence your hunger and satiety levels via your hypothalamus. Okay. Insulin is released from the pancreas, specifically the B cells of the islets of Langerhans, and it is released in response to these two different duodenal hormones and also to the absorption of glucose into the blood. So high blood sugar triggers the release of insulin. So what exactly does insulin do? We go up here, it is distributed throughout the body, and insulin is going to bind to its tyrosine kinase receptor, which leads to a lot of different things, but the main story I want to tell here is that insulin is going to trigger the insertion of more GLUT4 glucose transporters. Okay? These ones are insulin dependent and greatly increases the uptake of glucose into different cells. In the liver, glucose enters via these GLUT4 transporters, and the insulin receptor is also going to trigger the process of glycogenesis with this glucose, so more storage of this molecule, and glycolysis, the anaerobic step in cellular respiration. So glycolysis essentially uses this glucose to make more cellular energy. Insulin is going to trigger the uptake of glucose into the adipose cells and further storage as adipose. Finally, insulin is going to trigger the insertion of more GLUT4 receptors or transporters into muscle membranes. Okay, so skeletal muscles as well as cardiac muscle, etc. And so the glucose is taken into these muscle cells. It is used for immediate energy, and it can be converted into glycogen and stored until the muscle needs it again. Now, this insulin, this mechanism, is not the only way that muscles, for example, can take up glucose. They have a baseline intake of glucose through other GLUT transporters, but in order to increase their activity level, they need to have these GLUT4 channels as well. There's one final hormone to mention. This is VIP, vasoactive intestinal peptide. This is going to be stimulated by proteins and other nutrients within that chyme. And essentially, the presence of any nutrients means that soon, right, farther down in the digestive tract, there's going to be this flood of monomers of different biomolecule classes into the blood, and therefore, this protein is signaling the blood vessels to widen and essentially deliver a lot more blood to the intestines, so that more of these nutrients can be absorbed and therefore used by the body. So vasoactive intestinal peptide is secreted in a lot of different places throughout the body, but in this case, food in the small intestine triggers more blood to go to the small intestine and therefore more absorptive power. Okay. Once again, this has been a summary of some of the hormonal mechanisms involved in the digestive system. Ghrelin is a hunger stimulant, so it's anorexic. Gastrin promotes stomach action. CCK triggers the release of enzymes and bile into the duodenum. Secretin, the release of buffers into the duodenum. GLP-1 triggers the release of insulin from the pancreas. It also, up here, is an anorexic molecule that talks to the hypothalamus. GIP triggers the release of insulin, but also leads to more bone deposition and storage of energy as adipose. Insulin, released from the pancreas, triggers the uptake of glucose by cells. right, is also released in response directly to high glucose in the blood. Insulin is anorexic. Insulin triggers more glucose uptake by cells throughout the body and triggers them to, or these molecules to be stored as glycogen or to be used to generate energy for the cell's function. This is particularly important in muscle cells and in the liver. Insulin can also trigger storage of glucose in fat and other cells. VIP, one more time, is going to increase the blood flow to the intestines by dilating the vessels. Thank you so much for watching.